Lipophilic hormones such as steroids and thyroid hormones pass easily through the cell membrane into the cells, where they bind with the intracellular receptors. Now, intracellular receptors are of two types: the nuclear receptors present inside the nucleus or the cytosol, and the IP3 receptors that are located on the endoplasmic reticulum. IP3 receptors comes under the second messenger system. They are actually linked with the calcium channels and release calcium in response to hormone binding with the membrane receptors. We have talked about this mechanism while discussing the membrane receptors. Now, nuclear receptors are more important to discuss here, as the lipophilic hormones directly bind to them. Nuclear receptors belong to the superfamily of DNA binding protein. That is the reason when they are activated by binding of hormones, they bind at a specific segment of DNA known as HRV. This binding leads to the transcription and protein synthesis. They are also called ligand-dependent transcription factors because they initiate transcription only when a ligand, for example hormone, binds to them. In other words, they have the ability to bind with the DNA even in the absence of ligands, though this binding cannot initiate transcription. Before going into the details, let's have a look at the structure of the nuclear receptors. Intracellular receptors have three main parts: amino terminus, DNA binding domain, and carboxy terminus. Amino terminus interact with the transcription machinery, whereas DNA binding domain binds with the DNA and carboxy terminus is the site for the hormone binding. Now, depending on the location and mechanism of action, nuclear receptors are classified into two main types: type 1 and type 2, also known as class 1 or class 2. Class 1 receptors are those present in the cytosol of the cells. In the absence of hormones, they are present in complex form with the heat shock proteins. When the hormone enters the cell and binds to class 1 receptor, HSP dissociates and the receptor hormone complex is translocated into the nucleus, where it binds to HREs, that is, hormone response element, on the DNA. This makes a nuclear receptor DNA complex, which then initiates transcription and makes protein. Examples of class 1 receptors are subfamily of glucocorticoid, progesterone, estrogen, and androgen receptors. Now, class 2 receptors are those present inside the nucleus. Their binding with the DNA does not depend upon the presence of hormones. When the hormones are not present, they often form complex with co-repressor proteins. It is a type of protein that represses the expression of genes. Hence, the complex cannot initiate the transcription. When hormone is present, co-repressor protein dissociates and hormone binds with the nuclear receptors. Additional proteins such as RNA polymerase are then recruited, that is then transcribed to the messenger RNA and proteins. Examples of class 2 receptors are the thyroid hormone receptors. Two other classes of nuclear receptors also exist, class 3 and class 4, but they are almost similar to class 1 and class 2 with minor differences and are of no importance while discussing hormones. So this was all about today's topic, hormones, mechanism of action. I hope it's clear now that how hormones bind with a different receptor and each receptor lead to a different mechanism of action and then ultimately produce the cellular response. If you have any query, comment down below and keep watching sqadia.com.